All right. Hi, everyone. I'm Katelyn, a scientist in Charlie Emerson's lab, and I've worked with Charlie for about almost 7 years now. And so some faces I recognize from one of the last patient meetings we had back in 2020. I met Kathy joining their meeting one time to talk about the research. So it's exciting to see some familiar faces and see some new faces too. Um, I'm really excited to talk to you guys about how we're using cell and animal models to do therapeutic development in the lab. I'm not going to get too much into the science, but if you have any questions, please interrupt me as you see fit. So as Larry was telling you, we have a lot of muscle biopsies in the Wellstone repository, and I won't go too much into that, but I'm going to talk about how we use those muscle biopsies to do therapeutic research in the lab and how you go from the biopsy to the laboratory. So when you have a muscle biopsy or an open muscle biopsy, they take out these cells and we can put them into a dish and we isolate what's called the muscle stem cells or myoblasts. And so these myoblasts, just to give you an idea of what this looks like in a dish, so this is a picture of some patient myoblasts that we have. And as Larry said, we have greater than 50 families, as we call them, or FSHD patients with a first-degree relative. And we can compare that FSHD patient and how the muscle cells act in a dish to the first-degree family member. And it's a really powerful tool that we have because a lot of times people just have patients and they don't have anything to compare it to. But when you have a family member, it's really more powerful to say this person is related to this person. What's different between the person who has FSHD and the person who does not have FSHD? And then, so I'm not going to talk much about this, what happens when you have a skin biopsy, but Dongsheng will in a little bit. So when you have a skin biopsy, you can isolate cells called fibroblasts. And this is a picture of what the fibroblast looks like in a dish growing out of the little, you chop up the skin into little pieces, you put it in a dish, and after a couple days, these cells called fibroblasts will grow out or come out of the skin sample. And we can isolate those fibroblasts and turn them back into iPSCs that Dongsheng is going to talk about a little bit later. I don't mostly focus on that. I focus on the muscle cells. So what do we use muscle cells and myoblasts or muscle stem cells for therapeutic development? So this is just an example of how we use cells in the lab to do therapeutic research and develop therapies. And I'm going to show this based on one of the therapeutics that I'm developing in the lab, which is an siRNA targeting DUX4. I'm not going to tell anybody about DUX4. I think you guys all know what DUX4 is at this point, so I'm not going to go into any of that background. But if you don't, you can come talk to me later and we'll have a fun discussion. So as I showed you some pictures of some myoblasts before, these are what the myoblasts look like growing in a dish. And then we can add therapeutics to them. In this case, it's an siRNA as the cells are dividing. And then we put them in a different kind of cocktail of cells and growth factors, and we take that out and you can turn them into muscle fibers or myotubes in a dish. And that's what you can see down here. You can see it looks different from these myoblasts growing, and then you differentiate them into myotubes or muscle cells in the dish. And so that's what they look like here. And then we can isolate these cells off of the dish and we can look at the expression of genes, so DUX4 genes or muscle differentiation genes, to see how the muscle is doing and see how the cells are responding to the treatment. So in this case, I've treated the muscle cells with two different drugs. So one is called ZU01 and one is called ZU07. And so if you look, the differences here, so it looks like this one doesn't look like it's turning, I call it swoopy. It looks swoopy. So you see these cells look like tiny little balls, and then as you differentiate them, they look swoopy and they swoop around the dish, and that's the differentiated muscles. This one looks less swoopy, let's call it. And so we can measure the swoopiness in terms of muscle differentiation genes and DUX4 target genes. So I'm going to do 3 graphs total, I promise, and it's just to make a point that our drugs are doing something. And I'll explain them all. And if they don't make sense, please ask me. So when we look at DUX4 target genes, so this is a gene that is activated after DUX4 is turned on in the muscle, because DUX4 is only expressed in 1 in 1,000 cells. So it's really hard to measure the amount of actual DUX4, but we're working on a program that can measure actual DUX4. So more on that later. And then we can see when we treat with these drugs, what happens to these DUX4 genes. And so big purple arrow showing when you treat with these 2 siRNAs, you knock down DUX4 target gene expression. However, you don't want to knock down the swoopiness, as I called it before. So this number 7 here knocked down DUX4 target genes, but it also stopped muscle from differentiating. So we don't want that. We want good muscle. We want muscle to differentiate and to be healthy. And so based on that information, we're going with this number 1. So you get good muscle and you get no DUX4. That's the goal. So then now that we have this siRNA that gets rid of the DUX4 but keeps the muscle nice and happy, how do we get it to the muscle? And there's several strategies that companies that are here today and myself are using to try to get these drugs to the muscle in the body. And so Avidity and Dyne are here. And so their strategy of what they're doing is they take an antibody that targets their drug and their siRNA, and it brings it to the muscle. And so I'm not going to talk much about that. I don't know if Avidity or Dyne are going to talk about that, but just different strategy. What we're doing in our lab is a lipid conjugated siRNA. And so a lipid is a little piece that we can conjugate to the end of our siRNA. And I'm not going to go into too much detail, but if you're interested, come find me later. I'm more happy to have a more in-depth discussion. And so our lipid is conjugated to the siRNA, and our collaborators here at UMass Chan in the RNA Therapeutics Institute have developed this lipid that specifically accumulates in the muscle. So we're targeting our siRNA to the muscle using this lipid that our collaborators have identified. And so now how do we test that this is getting to the muscle? So we need an in vivo model. So in our lab, in the Emerson lab, we take advantage of muscle regeneration and we use a mouse model. And I'll explain a little bit about what that means. So muscle regeneration, when you have an injury, these cells called satellite cells become activated. They multiply and then they form, this is what they look like in a dish, as you've seen before. They multiply and then they fuse together to create new muscle. And that looks swoopy. And you can see these multinucleated cells here. Each of these little black dots is a nucleus. They get together and form muscle fibers. And so we take advantage of this muscle regeneration phenomenon in a mouse. So we use this mouse model. And so there's these little white mice and they have no immune system. And so we get rid of their stem cells specifically in their legs. And then we do a muscle injury to start muscle regeneration process and to get rid of the mouse stem cells in the leg. Next, what we do is we take those cells that I just talked to you about that we can grow in a dish, we isolate them and we inject them into one specific muscle. If we use the tibialis anterior muscle, which is right about there in the mouse. And so we inject these patient myoblasts from into the leg of the mouse. And then over about 2 to 4 weeks, these cells, and then we treat the mouse with a therapeutic, and then we take out the muscle and we look to see how our therapeutic affected the human myoblasts in the mouse. So, sorry, my slide's not working. So how does this look in the leg of a mouse? So we're growing human muscle from these human-derived myoblasts in the leg of the mouse, and then we're treating it with therapeutics to see how the therapeutic affects the human muscle in the mouse. And so this is a picture of one of our xenografts, as we call it, and it's just a cross-section. So if you look at this muscle right here, if you cut it right there, and then you look down the body of the muscle, so each of these little red dots, or we call them donuts, is an individual human muscle fiber. And so these antibodies are only picking up human muscle. And so all the blue dots are the residual mouse tissue there, but all the red circles or red donuts are human muscle fibers that we've grown in the legs of these mice. And we can figure out how the therapeutics with our siRNA are affecting the human muscle. So now my second graph, or third graph, I guess, technically. So when we use our lipid conjugated siRNA to get the drug to the muscle, we have a lot of success. So we're seeing about, this is again a DUX4 target gene. And so on the bottom here is 3 different patients. So we've created xenografts with 3 different patient myoblast lines. And in all 3 of those 3 patients, we see about 50% decrease in DUX4 target gene expression, which we're really excited about. So we've developed this therapeutic and we're hoping to partner with a company that can take it to clinical trials, because as an academic lab, we need a lot more money in order to do that. So we're currently hunting for a partner to help us take this drug to the clinic, but we're really excited about where it's going. And so thank you to everyone. And really none of this could have been done without really generous patients and family members who've donated these myoblasts that make, or the muscle cells that make all of this therapeutic development possible that we do in the lab. So thank everybody in the community, FSHD Society, for helping us connect to the community, especially in our funding. And of course, Charlie, who's around here, that's Charlie. And Jing does all of the animal experiments with me. And Dongsheng is going to talk to you guys, I think, next about what he does with the skin biopsies that we generate in the lab as well. And of course, our collaborators are really intelligent chemists that help us to figure out how to get the drugs to the muscle. So it wouldn't have been possible without our chemist friends as well. And thank you for your attention. If there's any questions, I'd be happy to answer them, or we can chat later one-on-one in the breakout sessions.
What are the possible potential advantages of using that lipid
The lipid.
system.
Can you repeat the question?
What is the benefit of using the lipid delivery system versus an antibody delivery system? So one of the main advantages I would say is, so it's hard to make an antibody and to make it active. And so we can synthesize this lipid conjugated siRNA in a lab, and you can make it, a lot of it, really easily and quickly and inexpensively. So we think one benefit of the lipid, and again, I've never compared the two side by side, so I can't say if one is better than the other. I'm sure Avidity has ideas, and again, I see you smiling over there from Avidity. So I don't know your data, and so I don't know if it's hard to make the antibody conjugated oligos, but in my mind, it's probably easier to make, just synthesize a lot of this lipid conjugated oligo in a lab versus trying to have the antibody component. But I don't know how they compare for delivery, obviously.
Good question. I saw that you had a 50% reduction in the DUX4. What does that equate to in terms of the efficacy? In terms of like what does 50% mean?
What does 50% mean in like relevant, like does it make improved function with 50% knockdown of DUX4?
Like what percentage do you need for it to be?
That's a very good question. And so these clinical trials that are coming out now are just starting to understand that question. We don't know how much DUX4 to be, like how much knockdown is going to be the ticket to getting functional muscle recovery. Again, Avidity is talking later, so they can probably elaborate more on this, but they've had really a great first data coming out showing 50% knockdown in their target genes from their clinical trial. And it looks like they're seeing trending increases in functional movements. And again, I'm probably misquoting all of the exact data, but I'm really excited about the outcome of, so I've been following it very closely. But 50% seems to have trending increases in functional readouts as of right now. But again, more is coming. And we're just learning these things as people are going through clinical trials with DUX4 targeting therapeutics. So we're excited to see Avidity's first, obviously, and we're really excited to see how this data is going to come out. So we don't know. The answer is we don't know what you need. We're hoping to just knock down as much as we physically can. And we're working on modifying our dosing strategy to try to improve that 50%. And so the chemists that we're working with are also continuing to tinker with how the modifications they build into the siRNA backbone and the linker and the everything to try to make it knock down for as much as possible for as long as possible. And so the chemists are working really hard on trying to update and make a 2.0 version of this to improve that 50% knockdown. So that's in progress.
Could you start to answer that question looking at a functional mouse model?
A functional mouse model. So you mean like the flex mouse or the, yeah. So we're planning on doing those experiments. We haven't done it yet because our model is a xenograft model, and that's mostly what we've been focusing on. But in order to figure out how much you need, how much knockdown you need, and how long it will last, we're doing that in the mouse models. That's up next. Exactly. Because as much as we love doing the xenograft model, and it's only replacing one muscle, so you really can't see functional improvements in one muscle that's humanized if you treat it. That one muscle is not going to tell you if the mouse can run faster or better, or if it's having less muscle pathology. So we can't tell that with our model. So we're hoping to use one of those. And what we're talking about is that they have mouse models where you can turn on DUX4. So mice don't have DUX4. It's a human primate specific gene. So mice don't have it. And so you can have a model where you have a mouse and then you give it a drug and it turns on DUX4. And then you can watch the effects of DUX4 turning on in the mouse and therapeutically intervene and try to solve the muscle degeneration that these mice have. So that's what we're planning on doing, but we haven't done that yet. Any other questions?
So this is sort of a one-time slice in time, right? So there's no long-term. Does it continue, or does it lessen the percentage? You haven't gotten to that point yet, right?
So so we've looked at one point. So this is a 1 week post-treatment for these mice. We looked at one time point. So we're not sure if we looked 2 weeks or 3 weeks, if we'd get further decrease in our DUX4 target gene expression. We haven't looked yet. But our chemist friends have used this same technology targeting a different a different gene, and they see functional, they can redose it also, which is important. So you can give more of the drug another time to continue to knock down, which we haven't played around with multiple doses yet either. That is something that's on our radar. But yes, we've only looked at one time point. And so we're hoping if we modify the timeline or the chemistry that we can make it more stable, the drug more stable and last longer. So we don't know that answer yet.
For additional doses.
Right. So the nice thing about the siRNA as opposed to like an AAV or a virus delivery strategy is that you can redose. So people shouldn't develop an immune reaction to the siRNA. So you can give them doses, and our collaborators were giving their therapeutic every 2 weeks for up to, I think, 29 weeks. And there was no toxicity, there's no problems, no immune response in the mice for up to 29 weeks. And they had 95% knockdown of their target for 29 weeks. So we're hoping to use that new chemistry that they just developed and do that with our drug as well. So there's better, 2.0 is coming. This is just 1.0. We're working on making it better, but I don't have that data yet. We're getting in the lab and starting that up now.